Right, and I think I know our next speaker. It's my colleague Phil Orr, who's going to be talking to us today about what drives changes in bloom toxicity. Thank you.
All right, thank you, Michelle. I'd like to start off by saying, not with a title slide, but with a blank slide. And whether it be a bloom of 1,000 kilometres long of Anabaena circinalis in the Darling River in New South Wales, or perhaps a toxic bloom of Microcystis aeruginosa in Lake Taihu in China, or even a toxic bloom of Nodularia spumigena in the Baltic Sea, as water managers, and many of you are water managers, and I guess as scientists, this is a headline we don't really want to see. This was a toxic blue-green algal bloom in the Caloosahatchee River, which flows from Lake Okeechobee in Florida just a couple of weeks ago. And Lake Okeechobee is becoming Florida's Lake Taihu. So one of the things, where's the pointer? Here we go. One of the things water managers would like to know is how the toxicity of this bloom changes. We know that there are toxic and non-toxic strains within the bloom, but how does the toxicity here differ from the toxicity over here? And of those toxic species, what's driving the changes? So my talk is really what drives changes in bloom toxicity, and my co-authors are Gary Jones and Michelle Burford. So I'd like to go back through a little bit of a historical background, and 1981, a paper was published by Wayne Carmichael, and it was titled The Mosaic Nature of Toxic Blooms of Cyanobacteria. So this was the very first study that was ever done, comprehensive study of bloom toxicity distribution in a lake, and it was carried out in Hastings Lake in Canada. What they did is that they sampled various stations around the lake at various spatial distributions and various temporal distributions. So they sampled one day, came back a couple of days later, and they sampled and again a couple of weeks later, and then did the same thing the next year. And they measured things like the surface distribution of phytoplankton, and they also measured the toxicity of the water at each of these sites. Now you notice some of these sites are very close together, and some of them are quite disparate in distance across the lake. And what they found was that that toxicity in the lake varies very, very rapidly. So July the 9th, July the 15th, and July the 18th, we've got big changes in the toxicity at stations that are very close together and over very short time periods. So the biggest one, the biggest toxicity, or highest toxicity that they measured was nearly 22,000 mouse units in 10 litres of water. That's enough toxin to kill 22,000 mice by intraperitoneal injection in just 1 litre of water, in 10 litres of water. But they came back 3 days later, and at that same site there was nothing, no toxicity at all. So this sort of led to a little bit of dogma, and people started to ask what was it that was driving those toxicity changes. And they actually said on a single day, as well as on different days, population density, species composition and toxicity varied greatly from station to station, and on some days big differences occurred between stations that were only 10 metres apart. So that led researchers around the world really to start to wonder what drives those toxin production changes, what triggers these organisms to produce these toxins. And over the years, several assumptions were made about toxicity of microorganisms of these cyanobacteria. One of them being that cells are equally toxic under a given set of environmental conditions. So you can provide a certain set of environmental conditions and you'll produce a certain toxicity of those cells. We know that toxicity does vary from non-toxic to highly toxic depending on conditions and what was driving those changes. but particularly what were those variables and what was the mechanism that was causing those variables to produce those cells or induce those cells to start producing those toxins and to do it so quickly. So a number of physiology experiments were carried out over the years, and this is just a selection of a couple of them to show you the wide range of studies that have been done. Van der Westhuizen and Elof in South Africa were studying the effect of pH and of culture medium on the production of microcystin by Microcystis aeruginosa. Peter Hawkins with Andrew Humpage and Ian Falconer were looking at cylindrospermopsin production by Cylindrospermopsis during batch culture and looking at various strains and under various conditions, particularly nitrate limitation. Karina Sivonen produced a classic paper in 1990 where she looked at the effect of light, temperature, nitrate, orthophosphate on a number of different strains of Oscillatoria agardii producing microcystins. And Jaakko Rapala in that same lab looked at Anabaena species and at those same growth stimuli looking at production of microcystins as well. But it wasn't only at the physiological level, it was also at the molecular level, and people like Melanie Kaepernick working in Brett Nyland's lab right here in the University of New South Wales was looking at the response of the transcription of the microcystin genes to various environmental variables, including light, and how that affects transcription of the microcystin biosynthesis gene cluster. So the only thing that was that you could say about any of those studies, the only thing that was consistent was that nothing was consistent. Everything was different. We got, oops, what have I done? Here we go. We had highly disparate results from all of those studies over 40 or 50 years, and nothing stood out as being a particular driver for any of those studies. But I think Melanie Kaepernick got it right when she said, our findings show that the effect of light on microcystin synthetase production is due to light quality and is initiated at certain threshold intensities, which are not necessarily reflected by observed intracellular toxin bioactivity. So there was no correlation between the transcription of the gene and the presence of the amount of microcystin that was present in the culture that they were looking at. Then at the International Toxic Cyanobacteria Conference in North Carolina in the mid-90s, I think, correct me if I'm wrong, Ian Falconer, there was some suggestion that toxicity might be related to growth, but nobody knew how. That was a sort of the discussion that was going around at the time. And more importantly, had researchers been measuring the correct parameters? That was pretty crucial. So there's a number of ways that you can measure toxicity, and a lot of those previous papers actually measured toxin per unit dry weight. So Sivonen, Rapala, Van der Westhuizen and Elof. Hans Utkillen and Nina Golm actually measured it relative to protein. Suzanne Lick has measured it relative to chlorophyll, and Don Anderson measuring saxitoxin production by dinoflagellates, and Gary Jones and myself and Suzanne Lick measured it by both unit of volume and toxin per cell. So there are all these different methods that you can measure toxin production and express it. Now we know that as we do a culture, is that going to work? Okay, over time our culture will get more toxic, the cell concentration will increase, but we also know that toxin increases. But what's actually going on in here? Do we actually know what's happening? We're getting more cells, we're getting more toxin, but what's that process that's going on in there? So we can look at it from an observational level, but we can also look at it from a cellular level. And over time, we've got one cell and it's going to become maybe more toxic with more toxin in it. But it's quite likely that actually we've got two cells, each with the same amount of toxin in it as the original cell. So Gary Jones and I did an experiment which we published back in 1998, and we looked at the production of toxin, microcystin in this case here, over time in a culture where we looked at corresponding increase in cell concentration. Now you'll notice I've expressed cells here as cells per mil and toxin here as micrograms per mil, so they're both expressed on a volumetric basis. And the stunning thing is that these set of curves, which correspond to different nitrate treatments, looks remarkably similar to this curve here. So as we're producing new cells, we're also producing more toxin. To take that a little further, if we measure the time period between two of our harvests, harvest 2 to harvest 3 in this case, and measure the slope of that line, because this is on a log scale, that slope corresponds to the first order rate constant on the of an exponential, of a first order equation. So the concentration at time 2 equals the concentration at time 1 times e to the rate multiplied by the time. So the slope of that line is pretty similar to the slope of that line. And if we have a look at all the other time intervals that correspond to our sampling of our culture over here for microcystin with our sampling for cells, we can plot the slope of this line against the slope of this line. So this line here represents mu tox, or the rate of production of toxin, and this line here represents mu c, or the rate of production of new cells. So what's the possibility of the various relationships between mu c and mu tox? Okay, if mu tox is less than mu c, it means that the daughter cells will contain less toxin than the parent cells. So the existing toxin is being reduced per cell, it's being diluted into a new population of cells. If mu tox equals c, then the parent and daughter cells contain equal amounts of the toxin, and that's said to be constitutive production. So it doesn't matter what you do to the cell, each cell is always going to contain the same amount of toxin. And again, if mu tox is greater than mu c, it means the daughter cells are going to contain more toxin than the parent cells. So just looking at those graphs that I had before, those 3 arrows represent the points on the line where we're plotting the mu c against mu tox, in this case microcystin. And the thing you'll notice is that there's quite a straight line relating the specific cell division rate and the specific microcystin production rate. That line has a slope of 1 and it passes through the origin. So what it's saying is as the rate of growth of production of new cells decreases, so the rate of toxin gets reduced as well. And I just want to be clear about something. When I'm talking about the rate of production, I'm talking about the relative rate. So going from 1 cell to 2 cells, going from 1 lot of toxin to 2 lot of toxin. So the rate of production of toxin and cells in both these strains, if you like, is exactly the same. I'm not talking about the amount of toxin, I'm talking about the relative rate, or the doubling time, if you like. So let's just quickly go through and work out what's happening here. So we can work out when the toxin is being produced by looking at the cell quota of the toxin, the amount of toxin in a single cell. So at the start, Q tox equals 1, the cellular quota of toxin equals 1. Over time, providing that Q tox is the same, sorry, let me say that again. If Q tox equals 1 in the daughter cells, then the toxin is being produced at the time of cell division. There's no change in the amount of toxin in an individual cell. If toxin is produced before cell division, then the amount of toxin in a single cell is going to increase, and then you'll get cell division after that. But during that time, the toxin quota within the cell will be double what the original parent cell is. And eventually it will change, the cell will divide, and Q tox will go back to 1. The other alternative is it could be produced after cell division, in which case Q tox in the daughter cells, Q tox in the daughter cells will be half the original toxin cell quota in the parent cell. and toxin will be produced, and again it will rise up to the same level as here. Qtox will again equal 1, but of course it can then continue to be produced and it can become more toxic over time, in which case we say that the metabolite, in this case the toxin, is a secondary metabolite because it's not produced in response to growth. So when a cell divides, a new quota of toxin is produced, and we can tell when that toxin is produced depending on what the Q value of that particular culture is. We know that Q will range from over a fourfold range from 0.5 to 2 times, and we actually saw that in the previous talk where we're looking at the toxin content of a cell, and it was ranging over a fourfold range depending on iron that was in the culture. But what we do know is that different strains can have Qtox that can vary by more than 100-fold. So you'll have toxic strains, very toxic strains, and some strains which have absolutely no toxin at all. So if we go back quickly now and have a look at some of the results from those previous studies that I mentioned, and we plot, we plot specific growth rate and against the microsystem production rate for the work that was presented by Sivanen on oscillatoria under N&P limited cultures, temperature and light. Again, we get a 1-to-1 straight line, and it doesn't matter what the strain is, what the environmental variable is, they all lie on that one line. Again, for Anabina flossaculi, N&P limited cultures, temperature and light, the relationship's not quite so strong, but it's still there. For microcystis aeruginosus, this is the data I put up before, we use 2 strains, an exenic and a non-exenic strain. Again, a 1-to-1 correlation between the cell division rate and the microsystem production rate. Anasuya Willis, who's in the audience here and will be talking later on, did a series of studies in a coordinated project with a number of other people, looking at Raphidiopsis raceborskii, which incidentally is the new name for Cylindrospermopsis. And again, it doesn't matter whether it was multiple strains or what the limiting nutrient was, still lies on that 1-to-1 correlation between the 0.5 and 2 that we were talking about before. Peter Hawkins on, again, with Cylindrospermopsis, again, a 1-to-1 correlation. And the only one that we really haven't talked about is Raphidiopsis raceborskii and production of saxitoxins in relation to CO2 metals and a whole range of other things that we might want to... all I can say about that is watch this space, because we're now working on that. So how do environmental factors drive changes in bloom toxicity? Well, the answer is that they drive it through the effect they have on changes in strain dominance, and not by triggering the toxin production pathway in the cell itself. And I'd just like to thank a number of people who have been on this journey, which is about a 20-year journey, I guess. Anasuya Willis and Tim Davis, who did a lot of the work on Cylindrospermopsin, Mattia Pierangelini and John Beadle, who worked at Monash University on Cylindrospermopsin production, and Cheryl Orr, who was a research technician in our lab in Griffith, Australian Rivers Institute, CSIRO Land and Water, and SEQ Water. And I just made it.
Do you have time for one question?
Thanks for that, Phil. That was a great talk. So, you know, when you're doing lab studies, obviously you're either agitating the culture or you're not. But in the environment, you have a mixed water column and then you get surfacing and scum formation and pile up through the wind and so on. I'm wondering, has anyone done any studies looking at growth rates in the scum?
Generally speaking, once you get to a scum situation, it's in stationary phase. So, yeah, but in a bloom situation, it's such a dynamic thing that it would be very difficult to do. People have looked at the growth in the water column, but not necessarily in the scum that I'm aware of. Yeah.
It'd be very tricky to do, but if there was no growth, then essentially all the toxin production's already done and dusted, and it's just the surfacing of the...
Well, any cultures or any samples that we've looked at, we've never seen an intracellular toxin concentration vary by more than about 3 or 4-fold. Doesn't matter what the strain is, doesn't matter what the environmental variable is, it always varies by about 3 or 4-fold, and yet people say blooms vary in their toxicity by hundreds or thousands of fold. The only thing that we can put that down to is a change in strain dominance, because there's been no recorded cases that we've looked at where the cellular toxin content of an individual cell varies by more than 3 or 4-fold. And that was explained by the division process. Yeah.